In this second module, devoted to perception, we'll look at the biology of the human eye and how that influences the way we can build an effective visualization environment. Our reference in the discussion of this topic is going to be this book called Information Visualization Perception for Design. It's a very good book, and I highly recommend looking at this for a deeper discussion on the topic. And you can easily find it on Amazon, for example. And and we look at this from the perspective of the entire pipeline of generating visualizations, how the image that is generated on the screen it's transmitted through light to the human eye, how then the human eye convert back the images into signal on the receptors on the retina, and then how the retina actually then send the information to the brain, and then the brain process the data, builds further patterns and abstraction so that it builds models of the world around. And so we see this kind of two concurrent processes. One is the bottom up turning light into patterns and increasingly higher level of abstractions. And then a top down where the models created by the brain can guide the human eye in understanding and scanning the visual space to get better information where and when is required through attention and other way of scanning the space. And so through the discussion, we'll see how the biology of the human visual system require this dual process, in a way how the visual system is fundamentally different than like a camera used to shoot a movie or even this particular video. So the author of the book also takes a very interesting perspective. I think it's a very good one where we, by having a deep understanding of the visual system, by visual system I don't mean just the eye, but the entire pipeline that involves eye and human brain and feedback, that can help creating a better interactive visualization environment, because we full understanding the dynamic of the information flow, not just how the light propagates in space. So the focus is on the human eye, and we'll start first from some low-level biological perspective, and then we see how that goes up a different level and influence the way a visualization environment can be designed and can be optimized. So the human eye kind of here is represented in a schematic way. We see the different components where we have the cornea, the pupil, and then the lenses. And this is kind of the area where the lights enter the eye and then goes through the vitreous body all the way to hitting the retina on the opposite side. This is kind of the more mechanical part of the eye, still very important. For example, you have to focus the light properly and so on. And then once the light reaches the retina, then the sensors start converting the light into signals that have to be conveyed and processed by the brain. So in this view, we see kind of the mechanical aspects of the human eye, for example, where the muscles help deform the lenses, the pupil can receive more light or less light. And also the interesting part is that the blind spot that is derived by the presence of the optical nerve in the eye. It's a key aspect, but also creates these regions where there is no sensitivity with respect to the light. So looking further detail, so here is the idea of light coming from the left to the right, and then after proper adjustment of the lenses and so on, the light will hit the retina. Now the retina is populated by a large number of sensors. Those are called cones and rods, and those sensors are actually not distributed in a uniform way. We'll see more about that. Here we have the optical nerve that I was mentioning before. And the different sensors have different sensitivity to frequencies of the light, and together can help building both a description of the shapes and also the color that are present in the particular scene we're looking at. So looking at the photoreceptors more in detail, the rods are the receptors that actually are more abundant. We have about 100 to 150 million of those distributed through the retina. They're distributed not in a uniform way. We'll see more in detail how that works. And they tend to be sensitive to low level of light and not really to colors. And so those, for example, are very useful when we're looking at things during the night, or generally when there is a low level of lighting condition. The cones are instead the sensors that help us distinguish colors. Interesting enough, there are not that many, right? There are 100 to 150 million rods. There are only 6 to 7 million cones. Not only that, they are not even distributed in a uniform way. We'll see more in detail. But more importantly, they are divided in three types: one that perceive red light, one that perceive green, and one that perceive blue. And this is the characteristic of the human eye. Animal different species actually can have a different number of such sensors. There are animals that see kind of just in black and white, so they don't have this variety. But there are animals that can perceive I believe, up to 12 different colors, so they would have 12 different sensors. So think about then all the variation of mixing those colors. It's kind of really hard to imagine the level of variety and also the complexity, how much of the brain has to be devoted to processing that kind of information. But different evolutionary characteristics have created a variety of visual system. Although the general structure is like this, it's just that the number of sensors and the distribution can be different in different animals. In this image, we see a high resolution photograph of the actual rods and cones. So the rods are this bigger sensor we see kind of in the back of the scene, and here in the front we see the cones. And of course, cones now, as I said, are different varieties. In the image on the right, we see the distribution of the cones by color, and you see also how non-uniform the distribution is, and also how the cones are put in a pattern that is not as regular as one might expect. But again, the human brain then is, given these irregular patterns, is able to reconstruct the images as we know we have in our mental images. So in terms of sensitivity to light, So the red cones are the ones that are sensitive to the higher frequency light. So here more on the right. And there are about 64% of all the cones, so more than half. Um, and that's kind of an interesting disparity. 32%, okay, so almost half of the red cones are green. And here you see the spectrum of the sensitivity to light and how the red and green actually overlap with each other pretty heavily. And maybe that's one reason why having a large number of them is important, so that one can resolve this difference. The blue cone are a much lower percentage. We're talking about maybe 4 to 5%, maybe a little bit more. There is always variability, but their spectrum does not overlap very much with the others. And so the human eye is able to get by with much fewer of them, but maybe also that leads to a lower ability to process that type of information. So if we look at the cross section of a human eye, so this can be reduced to a circle. And now we can then use the angle with respect to the center of the eye to map the distribution of cones and rods along the retina. In particular, we think we center this in the fovea, where there is a higher concentration of the color-sensitive cones, and then we go to the left and to the right. So that can be unfolded in this one-dimensional plot. At the center here, we see this area with the high spike, the red line of concentration of cones, and then there are very, very few cones as we move around the eye to the left and to the right. And then the rods, the blue line in the plot, they go very high concentration as soon as we get out of the fovea, and then they start decreasing in concentration as we go to the margin of the eye. Interesting is the asymmetry, where on the right side of this plot we see the gap created by the optical nerve when there is no sensors. And so this is the area that I mentioned before about the blind spot. It's something that we're not aware of because in a way the brain filled the gap, and the way we move the eye allow us to scan the scene and not make it into a big problem. But practically it's interesting that there is an area where there is total lack of sensor, and it's not too far in the periphery. It's actually relatively close to the middle, where you would have to have a high concentration of rods. So looking at the shape and the distribution of the sensors in the retina, we see again here the fovea, and here on the top left we see the image of the distribution of the sensor of the fovea itself. And so here we have a high density of cones, effectively only cones. And then as we go farther away to the periphery, we see instead a different distribution, where there is a high density of rods and only very few cones sparse on the retina. And here in this histogram, again of the perception of light, we see how the various blend of colors, for example, can be perceived by having the overlap between green and red. And so in this region where both sensors receive enough light and they're reacting to the light, we interpret the information neither as green nor as red, but as yellow. And so in general, we have this non-linear combination of the color. And again, this is a whole separate topic, but we need to be very careful that we don't, even though we really have just 3 sensors, when we resolve the color, there are additional color, and they do not emerge by just linear combination of the value of the other 3. Uh, Also, as any biological structure, the human eye come with all kind of variations. In particular, on the left, we see a typical eye with distribution of all 3 type of color sensitive elements. Interestingly, at the center, the blue cones are not present, and there is only red and green. On the right, we see the human eye subject to color blindness. And by the way, I have a certain degree of color blindness myself. I don't think this is the type that affects me, but where you can see that it's really physically the different distribution of sensors. In this case, it looks like the red cones are completely lacking, and that leads to a completely different perception of the images that one put on the screen. Now, there is a kind of a broad spectrum of color blindness, and it's important to realize actually that they affect a fairly significant segment of the population. So when we build a visualization environment and want to make sure that it is effective with everybody, for example, if we do a visualization that is for quick decision making, we need to be careful and make sure that the effect of color blindness may not lead to misinterpretation of the data on the screen. And so there are tools that allow to simulate the effect of color blindness given an image. And so depending on the particular application, it might be important to resort to such tools to make sure that we understand how the entire population can perceive the images that we create on the screen. Now, when we go from the physical color to the actual and sensors to the actual information perceived by the brain, we have to realize that not only the perceptors, the photoreceptors are non-uniformly distributed in the brain, in the retina itself, but the way they connect to the brain is also non-uniform. And so we have this kind of notion of a brain pixel, which really means what is the unit of information, basically the information that travels, say, on a single axon, a single neuron, that connected the eye, the physical sensors in the eye to the actual visual cortex in the brain. So the practical perspective is that if we think about this region as kind of the retina, and so the central part is where there's a lot of color sensitive photoreceptors, is also a very high density of neurons connecting to them, so all the information is actually very well communicated to the brain. As we go to the periphery of the eye, farther and farther away from the center, instead a cluster of photoreceptor can be connected to the same neuron, so that even though we might physically perceive a certain set of pixels, practical information gets averaged, and the brain can only perceive low quality information, irrespectively of the level of detail that physically the eye was able to resolve. So if we are at the very edge of the visual field. For example, at an arm length, we can really barely see an object that could be the size of a fist, so pretty big. While if we are really at the center of the retina, where there is a high density of photoreceptors, we can resolve about 100 points of the on the head of a pin, even at arm length from the eye. So very, very high density in the area of the fovea, where the not only the perception is high in the physical sense, all the information gets communicated to the brain, and the brain can process it very effectively. So overall, we see this effect that in terms of processing power, half of the work is totally devoted in the center of the foeva, and so, or in this part called parafoeva. So there is an area with high density connection to the brain, and half of our processing power devoted to the visual system focus only on that part. The rest is distributed non-uniform throughout the rest of the retina. So the outcome is that, for example, if we have a small screen nicely centered in the visual system, the pixels in the middle actually could be seen by multiple sensors, and that information is actually fully propagated to the brain, so we would be able to see sub-pixel information. While on the same screen, the information on the periphery, we might see the average of several pixels, because the brain pixel is larger than the screen pixel. And that effect is emphasized even more when you have a large screen. We can have like pixels here, large pixels at the center, and for which we will have a large number of photoreceptors also connected to a large number of neurons in the brain. So they correspond to a single pixel could correspond to a large number of brain pixels, while information at the edge would have a lot of averaging of several pixels for a single brain pixel. And so this is important to keep in mind, because effectively when we make a visualization, for example, we should see that even if we draw a simple table of numbers, having the full table displayed in detail, or a display of the type that we see here, where we have a lot of detail in a small area, and let's say that that's the area that we're focusing our attention on, and then the information gets lower quality as it gets farther away and gets really blurry at the far end. Here, obviously, the effect is a little bit exaggerated just to make the point, but effectively, this image, this blurry image, if we had fully focused on this central region, would have the same effect on our visual system, including all the way to the brain, as if we had a fully detailed display of the same information. We wouldn't be able to take more information than this. This effect, for example, has been exploited. For example, here we have a video from an NVIDIA research group where we look at the two images from the two eyes. Let me replay from the beginning. So this is doing full resolution everywhere, and that's kind of expensive because we have to generate pixels for all the elements in the image. Now, if we look at those red rectangles as the focus of the eyes in this scene, I think about having a stereoscopic display like in a VR environment. Now, we could display high quality only the center of those rectangles and make the scene increasingly blurry as we go away from the center. So here this effect is shown with more stabilization, so that you don't have the kind of the blocky effect that was emerging before, but the same in a way computational requirements. And here improved even more, where elements far away they are displayed low resolution, but also the contrast and the edges of the different parts of the image are being exaggerated, so that our low quality perception of the images does not become a problem. And so the idea is that if we can track the location of attention of the eye, the same image can be rendered much faster, because the number of pixels that we need to generate is much lower. So just to quickly look at this is the original picture. You see how many pixels need to be generated. And then as soon as we switch to the focus on a location, you see how much data does not need to be generated. This is kind of very blocky image. And then, of course, one can just improve a little bit on how the image quality is generated, but effectively the cost is really the same as generating that blocky display shown before. This type of effect can be exploited also when we have a very large display, and so a user can have focus in a certain region, and then we see how if we have cameras that track the eyes of the user and determine what is the location of attention, then there is an area far away from the area of focus that can be really rendered as super low quality, saving rendering time and increasing the speed of the display and the interactivity of the visualization environment. So this can be set up in different situations, as long as we are fully aware of where the user or the users are looking at. This, though, also created a question, is that then if we have such a narrow view into the data, how can we quickly extrapolate and understand a large image with all the details? And the truth is that our human eye, again, is not like a camera that look at the scene. Is more like an element that scan a certain environment, looking for details all over the place. So in this image here on the right, we have this set of points and lines that correspond to the trajectory of the eye that start looking at scanning in different parts of the image. And although we have the illusion of seeing a picture, we actually continuously, actively scan different locations, different areas that seem to be more interesting. Again, that's where we have the feedback loop from the brain to the eye and guide this process, so that we can actually have high quality information when it's needed, and maybe we leave with low quality information when it's far away from the area of interest, and still expect to have a mental picture that is high quality. A typical example would be a scene like this, when there are two speakers, and again, we would not perceive immediately both. We will probably for a moment focus on one and see a face in detail, and the rest is kind of blurry. Then we scan and focus on the other speaker and compensate for that, and then blend the information to get in our mind a high quality picture on the entire scene. But we really physically, we never see that single image all at the same time. So this phenomenon is well studied, and this movement is called saccades. It's a French for jerk. It's this jerky movement of the eye, where we move say for 20 to 100 milliseconds, scanning the space for a location of interest, for example, where there might be a big gradient or some interesting features. And then suddenly we stop for like 200 to 600 milliseconds, resolve that particular area, and then move on and scan to a different location. And so the result is this kind of jerky movement of the eye. So here we see that there is a system that is tracking the location of the eye, and then study how we are scanning a space. And so looking at the direction of this allow to build effectively an image of this type that we see here, where those are all the location where the eye stop for a moment and then go on and look for a different area of interest in the image. And then by this long sequence of jerky movements, we build actually a stable image in our as a mental image, even though we still see all those small elements. So to conclude, we have been looking at how the physiology of the human eye influence the way we look at the visualization, and how important it is that we understand how that works. For example, for optimizing a rendering environment and visualizing only what's important, as long as we're able to track the eye movements. So we have the light that comes into the eye, then the light gets converted to a signal from the photoreceptors, but then those signals again are not uniformly transferred. Some are transferred in very high quality and high density, some are kind of averaged away, and then the human brain start building patterns and abstraction based on our priors. And then by knowing the building some model of the world environment that can guide back top down the attention, and then help scanning better and better the scene to build at the end a high quality result. So with this, we conclude the second module, and next time we'll go further in detail to understand how we deal with different phenomena like contrast in a scene and other perception aspect of the visualization process.